Hi guys, I'm Todd. Me and my teammate Wendeng is here to talk about our project, which is contrast enhancement prediction in brain MRI. For the past 3 months, we have been working with Professor Greg Zacharczyk, who is a professor of radiology at Stanford University, and Andrew Shaw, who is a researcher at University of San Francisco. Here is an image of MRI, and if I tell you that there is a tumor present in this image, it's pretty hard to tell where the tumor is. However, the task becomes much easier with this image because there is a white bright dot that is highly contrasted with the rest of the brain regions. And this contrast is achieved through the injection of gadolinium-based contrast agents. This is why that gadolinium-based contrast agents is widely used in MRI practices. However, the drawbacks of using it is that it could have harmful effects on human body. Therefore, in our research, we are trying to use deep learning algorithms to predict those contrast regions in the brain instead of relying on gadolinium-based contrast agents. 4 different MRI modalities to predict the final output. Each MRI modality contains a unique set of information about the same brain. And to get to the final predicted contrast enhanced MRI, which is also called mBravo GD, the GD stands for gadolinium, we are adding an intermediate step in there. Our model first tried to predict the residual between the contrast enhanced MRI and the non-enhanced MRI. And we're adding this residual to the original non-enhanced image to arrive at the final contrast enhanced MRI. We have about 500,000 slices of those non-enhanced images, and we have about 100,000 slices of the enhanced images. To improve the generality of our model, we're doing normalization, rotation, and flipping on the non-enhanced images to make sure that our model can generalize on images that have different orientations and on different scales.
This is Wendeng. Thanks, Todd, for the detailed explanation of our project's background and how did we pre-process the data. So as you know, the model that we selected is a basic UNet. The reason why that we choose UNet is that our goal is to provide a predicted mBravo GAD with the same spatial dimensions as our input image, and UNet can handle this problem well. In each layer, there are two 3x3 convolution with ReLU. In each downsampling layer, we use max pool with 2x2. In each upsampling layer, we use the 2x2 up convolution. Totally, there are 4 downsampling layers and 4 upsampling layers. In the last layer, there is a 1x1 convolution. The metrics that we selected to evaluate the performance of our model are PSNR and SSIM. For both of the two metrics, the higher score that you get, the better performance your model is. For PSNR, a good score usually located within 30 to 40. For SSIM, the score ranged from 0 to 1. For PSNR, we got 36 on the training dataset and 31 on the validation dataset. For SSIM, we got 0.94 on the training dataset and 0.83 on the validation dataset. Both of the two metrics means that our model performs good. Let's look at a set of results. We take patient ID 0428 as one of the test data. On the left corner, it is the predicted residue. We clip in negative value to 0 and get the clipped predicted residue. We add the clipped predicted residue with the true mBravo and get a predicted mBravo GAD. We can compare the predicted mBravo GAD with the true mBravo GAD. We can see that for the enhancement part, this part, we successfully predicted it. And that is what we are looking for. Here are some brief conclusions for the technology that we use in this project. The image augmentation, like rotation, prevents our model from overfitting. The adjacent slices helps our model to learn the spatial attributes. The additional modalities enables our model to learn more detailed information, and the residual helps our model to focus on the contrast part. Next steps. For the next steps, we would like to add more modalities like flare, DWI2, and etc. We would like to clip values smaller than 0 and remove outliers in true residue. We will stack the slices back together to get a whole brain volume in negative format. We will try to segment the scope fight. We would like to try some different architecture like GAN. We would like to segment the MRI image and do AOC and ROC, and we would like to train on more data. Thank you.